It is really fantastic to be honored here, first of all, at this amazing research institution, which has such a long history and distinguished sort of one of the first places that really spoke up for being a biomedical research institute and not just a hospital, sort of really speaking into the science that has become one of the greatest things that our country is contributing. The second has been to be recognized by the Helen Dean King Award, who's really someone, it's important to recognize these people who early on took the steps to open the doors that the rest of the women in science could go through. And she represents a great example of that. And I want to point out that the Wistar rat has been very important in neuroscience in my own field. And I'll mention again, it's one of the best models that people have been able to use for things like studying depression in animal models. So a little bit of a personal connection. And finally, it's been fantastic to spend the day with the scientists at Wistar, particularly the women who are scientists, some of the men too. And but it just, to connect, to hear about the work, to hear the enthusiasm, to learn about science that is different from my own. This is why we're here. And it's the excitement of learning about science and being with other scientists that keeps me going and keeps me coming into this enterprise every day. So what I want to talk to you about today is neuroscience. This is my love. I am a neuroscientist, and but first I'll give you a very brief reminder of who I am and how I got here. I grew up in Athens, Georgia, and attended the University of Georgia. I then went to MIT as a graduate student, where I worked with Bob Weinberg. There I worked on the genes that cause cancer, which is a topic that connects me with a number of the people here at the Wistar then and now. And in particular, I cloned, when I was a graduate student, a gene called NU for its role in neuroblastoma, which subsequently was shown to play a key role in the development of breast cancer in a subset of human patients by Dennis Slayman, and then was subsequently developed further by Genentech to develop the drug Herceptin, which was the first rationally designed cancer therapy that was designed to specifically target a genetic change that occurred in a cancer cell. So this was I'm a very small part of a great story. And that great story is how understanding the basic science that underlies disease processes can ultimately, through the work of many, many people over a decade or more, lead to the development of therapeutics. And it's that hope that I bring to neuroscience as well. So as a postdoc, I switched fields dramatically, but only moved across the street physically to work with Bob Horvitz, a brilliant geneticist, and started to study the nematode worm Caenorhabditis elegans. And I will try to explain to you why I thought that was a good idea, specifically to understand the biology of behavior, which fascinated me. And that led to a faculty position at UCSF, followed by a faculty position at Rockefeller University that I started in 2004. I've studied a variety of questions about the brain, like the genetics of how olfactory sensation works, the development of the nervous system, how nerve cells grow out, find their targets, and make connections. And finally, the main focus has been on neural circuits and behavior. Now, over time in your career, life is long. I was talking about this with some of the junior women here. You find yourself doing other things as well as the science that you started to do. And I have spent some time working on supporting science in other ways, in a way that I've really enjoyed. So for example, I was embedded at the NIH for a couple of years working on something called the Brain Initiative, designing new ways to study the brain that transcend what I can do in my own lab, but can instead benefit the field. And similarly, for a number of years, I spent half my time at Rockefeller and half my time at a philanthropy, the Chan Zuckerberg Initiative, again, to really support technology development much more broadly, and to support open research and collaborative research to create new models to advance science to the benefit of all fields. And one of the good things about life being long is that you get to participate in such different things. The science itself, the planning that the government does to try to create new scientific programs, philanthropy and technology and the tech sector. We kind of move through life in different domains, and that's really, again, a privilege that we have that I've enjoyed. So that's sort of my short story. And now here is my real question. I just think I can't understand why everyone is not a neuroscientist. I just think there's so many reasons to learn about the brain. It's a wonder of nature. It gives rise to a stunningly diverse range of perceptions and thoughts and memories and actions. It makes us who we are. And at the same time, it's an incredibly complex organ. It consists of 86 billion neurons. That's more stars than there are in the Milky Way. And these make 100 trillion connections with one another. So there's just this incredible network. It's the most complex object that we know about. And on the one hand, we want to understand it. On the other hand, we are daunted to some degree in understanding how these billions of nerve cells and trillions of connections generate complex and meaningful patterns of activity and how those are converted into behavior. But it's important that we do that, not just because we should understand ourselves, but also because we need to learn more about the brain, because it is not just our greatest strength, but also our greatest weakness. And it is the part of our lives that we are less able to mitigate when it goes wrong. So we are going through tragic disorders that strip older people of their abilities and capacities, like Alzheimer's disease and Parkinson's disease, which affect over 6 million people in the United States now. And that will only go up as our population ages if we don't develop better treatments. We have the thought and mood disorders like schizophrenia and depression and anxiety that represent psychiatric disorders. The neurodevelopmental disorders like autism and intellectual disability that prevent children from leading their best lives. And finally, many disorders caused by injury and insult, like post-traumatic stress disorder or stroke or addiction. We want to understand these. They represent abnormalities that affect the biology of the brain. And the more we know about the biology of the brain, the more avenues we get to addressing these very fundamental aspects of our lives. Now, these are very big questions. They're very complex questions, and they're fundamentally about us. I'm going to take a step back from them, moving really toward the psychiatric side of this, the psychological side of this. And what I want to tell you about today is some things that we've learned about motivation and emotion and the way that that functions in the brain. It's clearly relevant to mood disorders. It's an example of something that, does have real resonance in people's lives. And what it is a set of ways that the brain functions to reversibly and flexibly rewire itself to give rise to different behaviors in different circumstances. And this is a fun thing to talk about to a non-neuroscience audience, because you can relate to these kinds of problems in ways that each of us have experienced in our own lives. So it is not a surprise to any of you to hear that your brain functions differently when you're asleep than when you're awake, or when you're hungry, when you're in an irritable mood, when you're highly agitated, when you're in the presence of other individuals or seeking their presence, when you're in pain, and when you're in stress. These are all ongoing processes that occur in our own lives. We match different kinds of biological and social and psychological needs to our behaviors and our actions to create the right kind of response to a given environment. And what we've learned as a field over the past years has been that these are often under control of specific small molecules called neuromodulators and neuropeptides. These are, so the main way the brain functions in its sort of like fast, dynamic perception, action, memory mode is through electricity and chemistry that are occurring on incredibly fast time scales, milliseconds, sometimes microseconds. But these kinds of responses, hunger, sleep, these can be things that occur over minutes or hours. They represent long-lasting states of the brain, and they actually engage a different kind of chemistry in the brain. And that chemistry involves these molecules listed at the bottom. The neuromodulators, like dopamine and serotonin, and neuropeptides, like endorphins or oxytocin. And maybe one way to talk about sort of the significance of these molecules is that even lay people, a lot of lay people, will know maybe all. We'll know what dopamine is, or we'll know what oxytocin is. We'll have heard of that in some part of their experience, because they do relate much more to the kinds of experiences that we look at in life. And one statement about the importance of these molecules is that they represent the targets of most psychiatric drugs and most drugs of abuse. So we know somehow that these molecules are affecting our own perceptions, or we would not have an opioid epidemic of molecules that are acting by mimicking the endorphin system, or other kinds, or we would not have things like serotonin, what are called SSRIs, drugs like Prozac, that are used to treat and to ameliorate different emotional disorders. So the question I want to pose for you is, how are these molecules working, and how can we learn how they work? So what we can say is that they aren't having the same kind of super fast activities that the main pathways are using. They're not always made in the same places. They're often made deep within the brain and then sent out to many other kinds of parts of the brain, projected to the brain from these deep, ancient parts of the brain to the newer thinking parts of the brain like cortex. And our real question is, how do they work to change behavior, and how do they work to modify the brain's function? So this is what they are. Now, what's the how? So we, in my lab, and many scientists, approach this problem by using a particular formalism. And we know that, you will know at the Wistar that many people study cancer by studying it in non-human animals. What might come as a bit more of a surprise to you is that many people also study behavior and the brain by studying it in non-human animals. It might seem like they're more different, and that would be a bigger reach. But the fact is, if you're trying to understand biology, basically the vocabulary of biology, the tools biology uses to carry out its tasks, are genes. And genes are used, they're shared between humans and other animals. And this came as a shock, even to those of us who started to study simple animals with the idea, well, we might be able to study some of the genes, but maybe not all of them. It turns out that humans and other animals share 99% of their genes. We are not special because we have new genes. We are special because we use old genes in new ways and map them onto this huge brain, for example. And it turns out that even invertebrates, like insects and worms and snails, share 75% of the genes that humans have. So we have this common vocabulary for biology that we can use in many different settings. And I'm going to try to explain to you how that makes sense when we look at these animals and we think, well, they can't possibly be feeling the same things that we feel, or at least they're not responding the same way or thinking about these things the same way. How can this be true? How can this be right? And I just want to say that in discussing this with you, sometimes, we always say we stand on the shoulders of giants in science. And I would like to point out that Charles Darwin himself, for a major part of his career, studied the behavior of worms. He wrote this book called Through the Action of Worms with Observations on Their Habits. He spent more time writing this book than he spent writing The Origin of Species. And while they were earthworms, it does point out that this is something that clearly alerted his attention, and he wrote about it quite in an interesting and intelligent way. And it's worms that I'm going to talk about. So why do we study a simple worm to study the brain? And the answer is that it creates this amazing ability to uncover the brain's logic. It's a really simple animal. It's literally transparent. You are looking at a worm through a microscope, and you can see every cell in its body. You can see every tissue in that transparent animal. And that includes that you can see every single cell in the worm's brain, of which there are exactly 302, each of which has a unique name, each of which can be recognized in every single worm, each of which has certain genes that are active in it, that characterize it, and each of which has connections with other neurons that are, again, highly reproducible. So we can turn to this animal over and over and learn more and more. And let's just point out that, first of all, 302 is a lot easier to take apart than 86 billion. And second, that nobody in this room has exactly the same number of neurons of exactly the same types with exactly the same connections. That's how it should be. We're different. That's part of the wonder, but it does make it harder to study. So we want to study something where we can just kind of break the problem down one step at a time, come up with some principles, and then see how those can be applied in more complex systems. And despite the simplicity, this is an animal, and it does the stuff animals have to do. It senses its environment, it learns about it, it explores, it aggregates with other animals, and it mates with members of its species, and it shares many genes with humans. So I'm going to start by telling you about the kind of behaviors that we think of as truly biological. And those are social behaviors. So here's an example of something. You need to understand social behaviors in order to succeed as a species. So if you are here, a goose, and you're a baby goose, you have to imprint on your mother and follow her around and learn to swim and walk and explore through that interaction. If you are an elk, you might have to have a certain kind of aggressive ritual to decide who gets to mate. If you are a honeybee, you have to be able to do a waggle dance to teach all the other bees in the colony where the new food source is and send them out to that. So these are just incredible behaviors. And basically every animal knows its own kind, interacts with the members of its own species, modifies its behavior, and succeeds based on its interactions with its own species. And we, as humans, succeed from the moment of our birth based on how well we can influence and understand and interpret the behavior of others. This is part of our biology too. And since every animal can do this as soon as it's born or grows, without any instruction usually, that tells us that these are behaviors that are set by genes, that these must be shared by the individuals of a species so that they can have this common understanding. And there's a really strong reason to support that idea. So I'm going to talk today about sort of three things: social affiliation, and that's mating or reproduction, social flexibility, and the effects of social isolation. And I'm going to talk about those, focusing for most of them on my tiny friends, the worms. Here, two of them have been made fluorescent to make them easier to see. And here they're gathering together in a little group of about 10 or 15 to do whatever it is they do in that little group. In this case, they're feeding. So what do we know about social behavior? There's a very famous molecule involved in social behavior called oxytocin. And oxytocin is one of these small molecules, a neuropeptide, that's a modulator made deep within the brain that projects across the brain and also to the rest of the body. Oxytocin, when a female mammal gives birth, is released into her bloodstream. It causes the uterus to contract so the child is born. It causes the lactation response so that the offspring can be fed. And it causes the mother's brain to change so that she forms a bond with her offspring, and also that she becomes more aggressive to fight off other potential attacks to her offspring. So this is really a remarkable piece of biology. Why this is important for the survival of the species is absolutely clear. You know, these little guys come out pretty helpless. And in fact, you know, this is directly administered to about a quarter of all women in labor to potentiate the delivery of labor. So this is a molecule in the brain where its effects on the biology are really kind of visible to us. But so for a mouse, maybe you kind of get it, but like something that's pretty surprising is that there is an oxytocin in C. elegans, and it looks like pretty much just like mammalian oxytocin. And we set about a few years ago to try to ask what that molecule does. That was Jennifer Garrison, a postdoc in the lab. And what she found is that it was very selectively and specifically required for C. elegans reproduction behavior. It was specifically for mating type behaviors, and it would reversibly and flexibly rewire the behavior of animals during mating to allow mating to be successful. Now, this is not a complicated behavior. This, by comparison to the behavior of a mammal that will have to take care of its offspring for days or weeks or months, is very, very simple. But the basic idea of this being a common vocabulary, of this molecule being kind of dedicated and fixed to social behavior early in evolution, is demonstrated by this example and by other examples. So what we find if we look across the evolutionary tree is that there's a long-term relationship between oxytocin and related peptides and reproductive behaviors. And that can be caring for your young, which occurs in both ants and in rodents, for example, or it can be in various kinds of mating or pairing behaviors, which we see across different animals. And I've left the human blank here, but I just want to say that if in all these different animals, this molecule has these kinds of roles, I think we can say this is an example of a vocabulary for biology. This molecule, what it says in the language of each species is, here's another individual for social behavior. And there are other social behaviors that are different. It's not one size fits all. And so here's a second example. It's actually kind of the opposite. So here again, we have a small molecule. It's a different neuropeptide, small molecule like oxytocin. It's got a different receptor. And at some level, it has the exact opposite effect. So this is a molecule that suppresses aggregation behaviors and causes animals to be solitary. And variation in this molecule leads some strains to be social and some to be solitary. And this is also regulated by stress. In many different animals, including C. elegans, stress leads to aggregation. So for example, if you see sheep in a pasture, if a predator appears, they all cluster together. Aggregation is a defensive mechanism, and that's what we see here. And a variety of people in my lab have studied this behavior over the years to try to understand how they recognize each other. It turns out it's pheromones that they detect, not surprising, but also to try and go into a little more detail about what does the brain do with this information? I've just kind of pointed that there's a gene that does something, but how does that actually get translated into this pretty dramatic change in behavior in the brain? So what we know is that the animals are recognizing each other by sensing pheromones. And we define the pheromones, and then we could define exactly which neurons sense the pheromones. And we looked at those neurons, those sensory neurons, and they were exactly the same in both. And then we could look at the wiring of the whole worm brain and see where those sensory neurons went. And we could see that some of them connected to that the sensory neurons connected to an escape circuit, and some of the connections of the sensory neurons were to an approach circuit. But both of these circuits are present in every worm. Every worm has connections to avoid the pheromone and connections to approach the pheromone. And so we can trace that through here and see that these different kinds of behaviors are encoded in the fixed anatomy. And that makes sense because the anatomy stays fixed for a long period of time, but this behavior can switch very quickly. So what happens here is that the neuropeptide shuts off one of those two options. It comes in here and shuts off the approach circuit, thereby leading this antisocial peptide to lead to antisocial avoidance behavior. So what we see here, and when the peptide disappears, the animal can go back to aggregating again. So the peptide is made transiently in response to stress and other conditions. And what it does is basically let the worm change its mind. It's like, yes, all these connections are still here, but I'm going to quiet some of them down. I'm going to turn down the dial on one set of connections and let the other set take place. And then at the end of this particular stressful period, I'm going to turn the dial back up, and now we're going to allow aggregation. And now we're going to switch over to between aggregation and avoidance. So this is an example of something that would be very difficult to do in other systems because you wouldn't know where the pheromone was being sensed, exactly what neurons were being activated, exactly how they connected to downstream circuits. We're getting closer to that now. We're starting to get that in the fly. Sometime in the next 5 to 10 years, we'll have a pretty good map in the mouse, actually. But at the time that we did this, which was about 10 years ago, this was the only place that you could really answer this question. So then the third point I want to make, the third scientific point I want to make, is to switch to a different kind of question, and that is how experience changes the brain. So I gave you an example of a short-term change to the brain that experience can make, that experience can cause the animal to change its mind by transiently shutting off a set of the connections to allow other connections to dominate, and then being able to switch back. There are many ways in our own experiences that the brain changes. And an example that I think many of us are familiar with and have been thinking a lot about recently is the process of being sick, of being exposed to a pathogen like a virus or a bacterium. And that leads to very profound behavioral changes. It leads to fatigue, reduced appetite, sleepiness, decreased sociability, nausea, learning about what made you sick so that you would avoid it in the future. This is a very robust change in behavior. And it's something that, oddly enough, just sort of wasn't something that people thought of as an interesting thing to study in the brain. It was just like, oh, the body gets sick, well, then you get tired. It's like, turns out that's not the case. Turns out there are very specific parts of the brain that are involved in detecting the experience of sickness, detecting the activation of the immune system, and generating these behaviors. But we're still very early in figuring out what those are. And so recently in my lab, we've taken a lesson from the COVID pandemic and other earlier pandemics, and we've asked ourselves, like, what happens in an animal's brain when it gets sick? How does it change? And here's an example of what some of the actual behaviors are that change in a worm's brain when it gets sick. So here's an animal, here are a bunch of worms sort of happily running around on their favorite food. And they're eating that food and having a good time. And here are their siblings being exposed to a bacterium that made them sick. And as you can see, they have figured out that this is not where they want to be. They figure it out, they avoid the toxic food, as originally described by Nathalie Pujol in Marseille. But they do more than that, actually. They avoid the odor of the toxic food. They learn the odor of the food, and then they avoid it in the future. So this is an example of something that a worm can learn. This is an important life-saving piece of information. They can retain that odor memory for life. A student in my lab showed that if you train them right after they hatch, as adults, they still remember this is not the food for them. And work from Colleen Murphy's lab at Princeton has shown that they can actually transmit that information, that memory, to one generation of offspring so that they don't eat that food either. So this is a pretty good set of behavioral changes based on one environmental input. And so we followed this up and we asked what was important for this, and we looked very broadly through the nervous system of the worm, and what we found is that there was yet another neuropeptide, yet another word in this language of these neuropeptides and neuromodulators that was specifically required for the animals to learn to avoid the food. And what happens is that when the animal is exposed to a bacteria that makes it sick, it increases the expression of this neuropeptide. It increases the production and release of the neuropeptide, so more of it goes into the system, and then that drives aversion. And it does so actually by shutting off a set of the neurons that would otherwise be driving attraction, which are themselves releasing another neuromodulator. So it's sort of like whenever we look at one of these sort of reversible acquired behaviors, we see this sort of change in which we engage different neuropeptides. But in each case, it's a different molecule. The molecules that are used to detect sickness are different from the molecules that are used to detect other animals or that are used to regulate reproductive behavior. So this is a good example of a behavior that we think we can invest in to understand further. And in fact, my lab, over the course of years, has been characterizing a variety of these different modulatory molecules. And another lab, Dennis Kim's lab at MIT and now at Boston Children's, has also made important contributions in this area. And what we find is that there are a number of modulators and neuropeptides that drive these behavioral changes. So there's one set of molecules, and that includes serotonin, another famous molecule, and this neuropeptide that help the animal to avoid the bacteria and form a pathogen memory. And there are other molecules that help the animal to stay on the bacteria, have the opposite effect. So it's constantly weighing these things against each other. And this is another important principle that we're seeing. These things don't act alone. These things act in groups. It's almost like they are together determining, well, things are better, things are worse, this is a good environment, I'm staying here. And we start to see this summation across multiple molecules and across multiple neurons. This decision is being made. I told you that these things project broadly in the brain. Different parts of the brain are weighing these inputs and deciding what to do. So this is helping us to think it's complex. Even in the worm, this is a complex readout. As far as we know already, something like 50 different kinds of neurons are involved. Thank goodness it's just a worm. Imagine how many it is for you. But I want to kind of point out that particularly serotonin was one of the molecules we found that was involved in this behavior. And just last month, a paper came out from scientists at Penn across the street showing that serotonin is altered in COVID and in long COVID in human patients. So when human patients get sick with COVID, their levels of serotonin fall. It's associated with various kinds of metabolic changes in their intestine, with changes in their platelets, and ultimately with changes that go to the brain. And these are associated with the kinds of changes that we might think were important during different kinds of experiences of being sick. And importantly, people who have COVID have the serotonin drop and then recover. But the people who have long COVID, they have a serotonin drop and it stays down. They don't recover. So it seems to correlate with whether you have long COVID or not. And this paper finished with a statement like, we should do some clinical trials to figure out if the serotonin targeting antidepressants might have a role in long COVID. And like 1 week later, a completely different group working independently in the Netherlands published a year-long study in which they treated long COVID patients with antidepressants like Prozac and fluoxetine, and they got a truly dramatic improvement in their symptoms, in their sort of brain fog, fatigue, sickness symptoms, which is pretty much matching really well. I am astonished by these two papers. In fact, when the first one came out, I was like, this is too good to be true. This is not going to work. Then a week later, I read the next one. I'm like, it really looks as though you will be able to target an important, I don't even want to tell you how many of these patients responded because I still have trouble believing it. It's 90%. So, it really looks as though for some people who have long COVID, there will be ways of targeting exactly what's going wrong to help the recovery to occur. So finally, COVID has told us something else, returning to the theme about social behavior, and that is that COVID has taught us how hard it is to be isolated. And, many of us have had this experience of being isolated, of feeling really alienated, depressed, lonely, upset. And there are experiments in mice that show the same thing. There's some really dramatic experiments in mice that show that if you isolate a mouse for just 2 weeks, it comes out of it with its behavior dramatically changed. It has increased aggression, increased fear. It's really an irritable, upset mouse. And this, David Anderson's lab has shown, is associated with a dramatic upregulation of yet another one of these neuropeptides. Again, there's a very specific change in the brain of this mouse from a happy, group-housed mouse to a social isolation mouse that dramatically upregulates a particular small molecule that's then secreted through the brain and acts on regions of the mouse brain that we know are involved in aggression and fear. So in terms of understanding how behavior changes by experience, happily, this response is ultimately reversible. Put the mouse back in a group, and after a certain amount of time, it will go back to healthy mouse behavior. But we can see here that an experience, a purely psychological experience for this mouse, has led to changes in its brain that then lead to changes in its behavior. And these changes in its brain will have to change for that behavior to move back. And I think that's a real, I think that's something valuable for us to think about as we look at our own selves and our children recovering from COVID, and also perhaps as we think about the criminal justice system and what we do there. So this is the conclusion of my talk. That brain circuits can be flexibly rewired by neuromodulators, that the way the brain works is to have more connections than it ever uses. It has an overconnected map, and it uses a subset of those connections at any given time, depending on what neuromodulators are around. And that means it can rewire flexibly in response to all of these different conditions, and then lead to all of these different changes in behavior. So I want to close just by returning again to me as a person, because just for a minute or two, because I was asked by my colleagues at the Wistar to say a little bit about being a woman in science and what it takes to be a woman in science. So this was the first slide that I showed you. I actually added a little bit of a personal element to the end of it here as well. And what is it like to be a woman in science? What was it like to be Helen Dean King? Very different from what it is today. Things have gotten a lot better. But when I look at what, even in my own career, has happened. I look, for example, at my undergraduate mentors, and I want to say two things about them. The first is that they picked me out when I was kind of an awkward 17-year-old and seemed to think that there was something there that was worth nurturing and moving forward. And I just want to say, maybe to every scientist in this room, you never know how someone who's in your lab just for a summer will be influenced by your example, who will be, by your introducing them to someone else who then gives them a longer place to work, just by pushing them along their way, so that decades later I still remember these people with such warmth and appreciation. Likewise, my graduate mentor, Bob Weinberg, who is a mensch, and Bob Horvitz, my postdoctoral mentor, who is a brilliant and wonderful leader. Just mentorship is so important for people to get ahead. And then I want to point out, really strongly, that here are two women, one at UCSF, one at Brandeis, who are about 10 years older than I am, a little more. And these women really had to go through a lot to succeed in science. At that time, it was really hard to make it. And they were discriminated against in obvious and non-obvious ways. I mean, at the time that these two women went to college, women still weren't admitted to most of the Ivy League schools as undergraduates. It's, that was more recent than we think. And having them kind of, again, open the doors and then invite everyone else inside was so important in my own career. And again, it's sort of remembering that the kinds of mentors you have at different points in your career, they start to show you the examples of how people can be successful. They start to bring you along. And that really makes a difference. The second thing I think that makes a huge difference is peer mentorship. And in my case, I've sort of picked out people I've collaborated with over the years. People who are roughly my age that I might work on in different ways, including women and men, who just ended up being the people that I could go to when I had a problem, when I wanted to complain, when I needed a drink, and who've just really come from very different fields, who've taught me things I knew nothing about, like theory and chemistry. And just working with these people at different places or on the Brain Initiative has been so enriching. And another group are my own lab members, here shown over a 25-year period in different kinds of pictures. And, they say it's not the destination, it's the journey, but it's not the destination and it's not the journey. It's the people you work with along the way. And that's what really makes for success in science. And that's what I think, now as we have more people from more countries and more sexes and more experiences and more races in science, we're sort of creating that culture for a broader group of people. It's not finished yet. We still have work to do. But I think turning to each other as a source of the answers to these problems is where we will succeed. So with that, I will thank you again so much for this honor and your attention, and be happy to